Good morning, everyone, and welcome to this webinar, which is the latest series of events by IHI to demonstrate the impact of our projects. My name is Tekung Lim, Scientific Officer at IHI, and I will be the moderator for today's impact series on vaccine. Next, please. I'm very delighted to have excellent speakers with us today, and I would like to welcome and thank them warmly for being available for this webinar. I will introduce them in due time prior to their presentation. Next, please. This impact series on vaccine comes very timely as we celebrate today the World Immunization Day. According to the European Centre for Disease Control and Prevention, each year vaccination stops 2.7 million people worldwide from getting measles, 1 million from getting pertussis, and 2 million babies from getting tetanus. You can see in this graph the evolution of the measles vaccine coverage worldwide on the x-axis, and on the y-axis you have the measles cases worldwide, and there is a drop of cases along the vaccination coverage. Next. I'm very much looking forward to hearing about the outstanding result generated by IMI projects on vaccine. Specifically today, we will hear how our projects have facilitated innovations in vaccines development and effectiveness assessment, going from reducing animal testing towards assessing flu vaccine effectiveness by brand. Without further ado, I will give the floor to Professor Ole Olesen, who is the Executive Director of the European Vaccine Initiative, based in Germany. EVI is an independent non-profit research organization focusing on developing new and improved vaccines, as well as developing new tools and technology for global health. Next slide, please. Sorry. Oh, before giving the floor to Ole, sorry, I have forgotten this. We want this session to be interactive, so we invite you to use the chat functionality at the end of your screen. Ask any questions you would like to the speakers, as well as providing your comments. Remind you that this session is being recorded. The recording of this webinar will be posted on IHI website as well on our YouTube channel right at the end of the webinar. Next, please. Next, please. And now, so that we will hear about how in clinical trials, how to limit animal testing in vaccine development. And the floor is given to Professor Ole Olesen. Ole, the floor is yours.
Thank you very much, Tek, and thank you very much for this introduction. And I can only applaud you and IMI, IHI for taking this initiative to mark the World Immunization Day on really highlighting the importance of vaccines and innovation in vaccines, and giving this opportunity to present some of the results that we have obtained collectively through the grants and for the funding and support we got from IHI. So I'm here, of course, today only as a representative for the entire consortium that we have been working in, the VAC2VAC consortium. And as you can see here, the title of the VAC2VAC consortium is Vaccine Batch to Vaccine Batch Comparison by Consistency Testing, which I've translated for the purpose of this presentation here into how can we reduce the use of test animals through collaboration and innovation. If you go to the next slide, please. So just to frame the issue here is that we still today use a very large amount of experimental animals in vaccine research and medical research in general. So globally, more than 115 million animals are used each year in different type of medical research and experimentation. And if we look at the graph here on the right side, this is just the numbers from Germany, one of the largest, the largest EU country. As you know, you can see that the use of experimental animals is sort of somewhere between 2.5 million to 3 million per year, which is a very large number. There are some promising trends, though, as you can see if you compare with the dark blue graph, which showed the investments in medical research and health research, which has gone up over the year, although despite of this, the number of animals used has stayed more or less stable. But still, there is a very large number of animals used in experimental studies, and therefore a large policy aim and also ethical aim is to try to reduce this number. And this is what we have been working on in the VAC2VAC project, specifically reducing the number of animals used for quality control testing. And as you can also see here, around 750,000 animals are used within the EU every year just on quality control testing. So next slide, please. So in essence, our primary goal in the VAC2VAC project was to develop and validate tests and approaches where we could use non-animal methods to replace the use of experimental animals. And we have done this by applying the consistency approach. And I'll come back to a bit more in details what this means in let's say, more lay terms. And of course, with the ultimate aim to apply this to reduce the number of animals used in routine vaccine production. So next slide, please. So our consortium consisted of 23 partners that were distributed across 8 European countries. You can see them shown here in this slide. Our project ran from 2016 until 2022. And what is interesting is that we had a quite solid consortium in the sense that we had a large industry participation of vaccine manufacturers, both in the human and the veterinary field. We had relatively few academic partners actually, but what we had instead was we had a very solid participation of public research institutions, and importantly for what we were doing, we had the participation of national reference laboratories with links to national regulators in 5 EU countries, which really was instrumental for pulling through the aim of the VAC2VAC project. So this hybrid of industry reference labs and academic and NGOs really made a very solid project. Next slide, please. In terms of setup, we had sort of what I guess is not an unusual setup. We had 6 operational work packages looking at different aspects. Given the nature of the project here, there were work packages looking at physicochemical methods, immunochemical methods, cell-based assays, validation, and also advocacy and more promotion. And then to keep everything interactive, we had then a work package 7, which looked at the overall management integration of the different activities and making sure we were directed towards the goal of the project. So next slide, please. So I mentioned already that, let's say, the baseline for the project was to apply the consistency approach in replacing animal testing with non-animal methods. And the background here is that vaccine batches, so every time a new vaccine batch, a batch of new vaccines has been produced, then it must be released onto the market. This is the responsibility of regulators in the respective countries. And they do this according to what is stated in the relevant pharmacopoeias for the relevant products. And basically 2 main things have to be tested, namely the safety of the vaccine material to demonstrate that there are no contaminants or any toxins in the batch. And the second thing is the potency, that the vaccine that has been produced actually does something, so that it actually elicit an immune response that is at least similar to what has been tested and what has been proven for that product in clinical trials. Now, traditionally, this has been tested in an absolute terms, in the sense that each batch has been tested on its own merit, so to say. In the consistency approach, we go towards a more relative approach, where based on reference batches, we can investigate the properties, both the physiochemical, immunological, and physical qualities, properties of each batch, compare them with a standard batch, and based on that, determine whether the safety parameters and the potency parameters are adequate to release the approach to the vaccine batch. So next slide, please. So one of the traditional ways of testing safety, but also a test that have often been criticized, is the rapid pyrogen test, or RPT in short. So pyrogens are fever-inducing substances, and this is an old assay to measure fever-inducing substances that was developed already in the 1920s, so more than 100 years ago. This assay was developed. And basically what it does is that you take the substance you want to test, you inject it into rabbits, and you test whether the rabbits develop fever. So in its normal standard setup, you test a group of 3 rabbits and you see if they develop fever. If they don't, if they have only a change of less than 0.6 degrees, then you say normally everything is good. But if you see an increase in fever above this in just one of the animals, then you move on to additional testing in animals, in further rabbits. So this has sort of been the classical test of testing for pyrogens. And in 2015, which was the start of the VAC2VAC project, this test was extensively used within the EU, and more than 50,000 rabbits were used for testing pyrogens at that time. So despite, and this was done despite actually already in 2009, there was an alternative to the rabbit pyrogen test introduced in the European pharmacopoeia, namely the monocyte activation test. So next slide, please. So if we look what is actually stipulated in the European pharmacopoeia here on the right about the monocyte activation test, it says, which is underlined here, that the MAT, the monocyte activation test, is suitable as replacement for the rabbit pyrogen test after a product-specific validation. And this is often where the shortcoming has happened, because nobody has really had the urge or the encouragement to take the step to actually undertake product-specific validation of the test for specific vaccines. So this has often been, let's say, one of the stumbling blocks, what has slowed down the introduction of the MAT test to replace the rapid pyrogen test. So in VAC2VAC, we set out to change that by really using our efforts to validate the MAT test to replace the rapid pyrogen test for a specific vaccine, namely the tick-borne encephalitis virus vaccine. And just to say in a few words what the MAT test actually is about. So what it does is that instead of using animals, we use a cell-based assay where the assay typically is based on human peripheral blood mononuclear cells, PBMC in short. You incubate the cells with the test substance, and then you measure specific cytokines, pro-inflammatory cytokines, and use that as a proxy for fever-inducing substances. So next slide, please. So in our case, and as you say, most of this work was done by Eliana Coccia and Mailena Paula from the Instituto Superiore di Sanità in Rome. But the setup is, let's say, in its essence, fairly simple. You start, as the small figures show here, you start with PBMC, you incubate them with your test substance, which in our case was the tick-borne encephalitis vaccine, which is called Ensopur. You incubate it for about 22 hours, and then afterwards you measure using a normal ELISA assay how much IL-6 has been released. And you use that as a measurement for fever-inducing substances that may be present in the vaccine. So this is the setup, but of course the devil is always in the details with these things. So after the basic setup, the key thing is to make sure that this is validated, repeatable, and can be used with high precision repeatedly by anybody who applies it. So next slide, please. And this was what was done. Here we see first the validation was done by testing not just one vaccine lot, but testing several here, initially 3 different vaccine batches, and testing them on 4 different donors to make sure that there was really repeatability and consistency in the testing method. So I'm not going to go into the details here. We have quite some time restraint, but I would invite, if you're interested, to really read the publication that was made on this validation, which was published in Altex in 2020. So there you would find all the details of the assays. If you go to the next slide, please. Just in summary, not just testing different batches, but also testing on different days, different sessions here on 5 different sessions and by 2 different analysts to test really the consistency and repeatability. Again, I'm not going to go into the details here, but just to tell you that this worked out well in the end, and it was possible to set up a very robust and highly stable and repeated assay with high degree of repeatability. Go to the next slide, please. So after this was set up by the academic partner, then it was transferred to industry, to GSK, which was the product owner, which then undertook an internal validation, after which they applied for official approval of using the test for release of vaccine batches to the German regulator, where the original product had been registered. And this resulted in that the rapid pyrogen test for this particular vaccine was officially used and replaced by GSK, so that they removed the use of the rapid pyrogen test, instead used the MAT test. And this was later transferred to Bavarian Nordic, which took over the product from GSK. And by the time that this was introduced, GSK estimated that more than 100 rabbits annually were used by them for release of vaccine product using the rapid pyrogen test. So that means that the introduction of the MAT test saved at least 100 rabbits annually by this replacement. What has also happened is that this has been taken forward in the sense that the Italian delegation to the European Pharmacopoeia Committee has requested that officially in the Pharmacopoeia, the rapid pyrogen test for the tick-borne encephalitis vaccine is officially replaced by the MAT test. This has been sort of part of a movement. Of course, I'm not saying that Vac2Vac alone caused this, but I mean, definitely contributed to the bigger movement that the European Pharmacopoeia Commission has now officially in June 2021 set out the goal to remove entirely the use of the rapid pyrogen test in all products by 2026. And as part of this, and I think also a general movement, is also heartening to see that actually within the 5 years between 2015, the start of the Vac2Vac project, and 2020, the use of the rapid pyrogen test overall has decreased by almost half in Europe. Go to the next slide, please. This is just one of the success, the replacement of the rapid pyrogen test with MATs in the tick-borne encephalitis vaccine. Just want to highlight here that Vac2Vac has generated a number of other results that is also paving the way towards replacing the use of experimental animals. Some of them are listed here. We've developed an assay for tetanus neurotoxin. We've developed 7 ELISA assays. We've produced 12 monoclonal antibodies that can be used to further development of non-animal tests. And also we have engaged heavily in stakeholder engagement advocacy towards the replacement of animal tests with non-animal tests. And this really brings me to the next slide, which is the conclusion, saying that I think sometimes we are a bit overlooking the importance of this type of research that we have done in Vac2Vac, because the results, the work that has been done may not be the things that create headlines or is very spectacular, but it's definitely something that is hugely important, because it is something that is really very tangible, very concrete results that we can apply immediately. So I think this is something that should be applauded and not be forgotten. And I think also it's very noteworthy that probably by now the results we have produced in the Vac2Vac, partly by the replacement of the rapid pyrogen test, but also some of the other results, has already resulted in saving the lives of thousands of experimental animals. I think also what's important to note that this would not have been possible, let's say, with traditional research, because then it would probably not have happened. So I think it's important also here to applaud the vision and the idea of IMI and IHI to really catalyze and promote these type of collaborations between very different partners to come together and really work together and jointly create the results that has made this possible. So with this, I would like to thank you once again for giving the opportunity on behalf of Vac2Vac to present, and thank, of course, funders and most of all partners and collaborators. Thank you very much.
Thank you very much, Ole, for this excellent presentation showing how the tangible result from a project has contributed to changes of practices among the industry. So that's a very interesting result here that you showed. And for our next presentation, in vaccine presentable disease, we always expect the vaccine to protect the person vaccinated against the disease, while at the same time lowering the transmission of the disease in the community. But as you can see with the recent examples of different vaccines, this can be a real challenge. We can think of the vaccines against COVID. We can think about polio with the resurgence of long-forgotten disease in several countries. So I'm delighted to give the floor now to Professor Dimitri Giavatopoulos, who will present how Periscope has tackled this challenge. Professor Dimitri Devatlopoulos is an associate professor at the Radboud University Medical Center, where he leads a research group focused on the prevention and treatment of respiratory infections. Dimitri, the floor is yours.
Thank you, Tech, and delighted to be able to present an overview of the achievements of the Periscope project, especially on World Immunization Day. So today I will be discussing with you how Periscope has contributed to the accelerated development of next generation pertussis vaccines. So next slide, please. So pertussis, also known as whooping cough, is a disease that is caused by the bacterial pathogen Bordetella pertussis. And this bacterium, once it enters the respiratory tract, can bind to the respiratory epithelial surface and cause acute respiratory infection, which is characterized by uncontrollable coughing. Although the disease is often seen as a childhood disease, it can occur in all age groups. But it is most severe in infants, where also most of the mortality is seen. In contrast to many other bacterial pathogens, pertussis is really contagious. So it has an R0 of 12 to 17, which means is that in a susceptible population, an individual that is infected would on average infect between 12 and 17 other people, which it means it's one of the most infectious pathogens that we know. And transmission of this disease occurs via aerosolized respiratory droplets. Now, there are already two types of pertussis vaccines in use today. And if we go to the next slide, we can see how they have impacted pertussis disease. So whole cell vaccines are vaccines that are based on basically the whole bacterium that is then inactivated. And these vaccines were already in use quite early, and they were more broadly implemented in national immunization programs in the 40s and 1940s and 1950s, at least in high income countries. And although these vaccines were actually very effective in reducing infant mortality and severe disease, they were also quite reactogenic. And because of public concerns about their safety, a next generation of pertussis vaccines was developed, and these are the acellular pertussis vaccines. And these vaccines contain a number of pertussis proteins, basically. If we go to the next slide. So these vaccines were more broadly implemented in the 1990s and 2000s, and in fact, at this point, almost all of the high-income countries have replaced the original whole cell vaccine with acellular pertussis vaccines. What you can also see here is that this switch was also associated with an increase in pertussis disease. And we go to the next slide. So pertussis at the moment remains like a leading cause of infant mortality in some regions in the world, and these are particularly the regions where there is still no vaccination or under vaccination. But in addition, we're seeing a resurgence in several countries and population cohorts that up until now were not really considered at risk, even though they are vaccinated. What I'm not going to discuss today, but what we're also seeing is that there are Bordetella pertussis strains, like they are starting to circulate, that are not producing some of the antigens that are in the acellular pertussis vaccines. So whilst we're at the situation at the moment that these acellular pertussis vaccines are really important for protecting against infant mortality and severe pertussis disease, next slide, it is clear that third generation vaccines are needed that provide more durable protection against the disease, but in addition also against transmission. Slide, please. Now, there are several barriers that make licensure of next generation pertussis vaccines quite complex. So as I already mentioned, there are multiple licensed vaccines available. And not only that, they're also recommended in national immunization programs. So that means that a placebo-controlled phase 3 efficacy study is not ethical at the moment. And in combination with the fact is that the vaccines are widely used, we see a relatively low disease incidence, particularly shortly after vaccination. On top of that, pertussis outbreaks typically occur in cycles. Every 2 to 3 years, there's a cycle, and it doesn't always happen in the same region. So there are regional differences, making it very difficult to predict outbreaks of pertussis, and therefore also planning of phase 3 studies. There are no immunological correlates of protection, and together, all of this really make it complex, because at this point, phase 3 efficacy studies are simply not feasible. Go to the next slide. And this is why the Periscope Consortium was established. And the objectives of this consortium were to foster the expertise, increase capacity in Europe to evaluate the next generation of pertussis vaccines, to identify biomarkers of long-lasting protective immunity, so that these biomarkers could help in the development of new vaccines, and thirdly, to investigate the impact of maternal vaccination on the infant's immune response to pertussis vaccination. So the Periscope project was ran from 2016 to 2022, and was a public-private partnership between 20 public partners and 2 FPA partners. So slide, please. So within this project, we followed a human-centric approach, and we performed a series of clinical studies. So these include 3 maternal infant vaccination studies, including in Africa, but also in the UK and in Finland. We performed a large international booster vaccination study in 4 different age groups. We performed studies in whooping cough patients to collect their samples. There was a preclinical aspect to the project. And last but not least, we established a controlled human infection model for pertussis. Slide, please. So all of these clinical studies provided samples that could then be analyzed in technology platform that was available within the project, with the objective to identify biomarkers of protection. Slide, please. And these biomarkers of protection, I'm not going to go into detail about all of them, but you can find the details on the Periscope website. But these assays are very much focused on measuring the establishment of memory T cells, memory B cells against pertussis, but also multiple analyses of functional antibodies. So we know that antibodies protect, but these antibodies, the way that they protect can differ. So for example, they can neutralize the toxic activity of pertussis toxin, but they can also bind directly to the bacteria and kill them, or prevent binding of the bacteria to the respiratory epithelial surface, and thereby preventing infection. Slide, please. That brings me also to the next part, and I would like to highlight this because I think this is really a key element of the Periscope Consortium, which is the establishment of a controlled human infection model. Slide, please. So if we look at pertussis disease, it is the classical pertussis disease that we often see in children is characterized by 3 distinct stages. So after you get infected, it starts off with the catarrhal phase. And in the catarrhal phase, it's basically you get a runny nose. It's very generic symptoms, not much specific for pertussis. But then once you go into the paroxysmal stage, this is characterized by the typical coughing fits that you get. And then over a period of quite a long period of time, you slowly go into the convalescent stage where you recover from this coughing. But the coughing can last for a very, very long time. And this can be quite an unpleasant disease to have, even for adults and adolescents. Slide, please. Now, well, one of the discussions that we had, like when we were talking about setting up this model, is like, okay, well, what should this model then reflect? Can we establish a colonization model? Like, are we talking about a disease model? And one of the key elements here is like, because we know that antibiotics can help, but they only help if you give them quite early on in the infection process. If you're already in the clinical stages, and you're starting to cough, then antibiotics don't really work because this disease is mostly mediated by toxins and not by the bacterium itself at that time. Slide, please. So there were quite some considerations, like when we were talking about this controlled human infection model, and these concern, on the one hand, ethical aspects, and primarily, of course, the safety of the volunteers. What are the risks? Are there potential benefits to these volunteers? And on the other hand, there were feasibility aspects to consider. For instance, containment of the infection, because, like as I mentioned, pertussis is an infectious disease and quite contagious. But also about treatment and about choice of challenge strain, etc. Slide, please. So we applied multiple risk mitigation strategies to limit the risk of severe disease. And for example, like by excluding volunteers that had any kind of risk factor for developing severe disease. We selected a bacterial challenge strain that was typical of the currently circulating isolates, that was then produced under GMP conditions, so that we have a challenge agent that we could that was very well characterized. We started with a dose escalation study so that we could give the minimal dose that was necessary to induce infection, but not give so much as that these individuals would get really sick. And of course, we looked at escape medication. To limit the risk of transmission to research staff or to the public, the initial studies were done in a containment facility, which means that the volunteers were basically inpatient for 2 weeks. Next slide, please. So this is the dose escalation study, and so this is just an overview of how this study looked like. You can find the publication online, so there's the reference here. So after the screening of volunteers, they get a baseline visit, and then they're admitted to the ward at the challenge day, and then they're inoculated intranasally with Bordetella pertussis bacteria, and then over a period of 16 days, they're quite intensely followed, both clinically but also looking for microbiology and immunology. And then at day 14, they're treated with antibiotics, and after 2 days, they're discharged. Slide, please. So this is like the way that this looks like. So in the inoculation, we have, as I said, GMP-produced bacteria that are then thawed and then diluted to the target dose, and this is how it looks like when you infect a volunteer with a bacterial inoculum. And then the remainder is investigated for to assess okay, how many bacteria were actually given, and are there any contaminants in there. Next slide, please. And this is how volunteers are then evaluated by a nasal lavage to look for the presence of bacteria in their nasopharynx, to see whether the inoculation actually resulted in an infection or not. Slide, please. So we started off with quite a low dose, but then we gradually escalated this dose, and then leading to an inoculation dose of 10 to the 5 colony-forming units of Bordetella pertussis, And that, in the end, resulted in infection of 80% of the participants. And so this was considered to be the standard inoculum dose. Slide, please. Because it was so infectious, we also investigated bacterial shedding, and over 6,000 samples from the environment were analyzed, from exhaled breath to contact surface, and that was in the end resulted in that there was no samples that actually tested positive for Bordetella pertussis, suggesting that at least in this model there is no environmental shedding. Next slide. So to summarize this, we established a controlled human infection model that induces asymptomatic colonization with Bordetella pertussis that is safe to use. We showed that azithromycin clears bacterial carriage, and there was no environmental shedding. And just to give you perspective, like this model has now already been used to test in a phase 2 study a candidate pertussis vaccine for pertussis. Slide, please. So the way that the vaccines are being put on the market, is when you have a preclinical product and you go into phase 1 and then phase 2 and phase 3. This is the classical licensure pathway. But at each of these moments, product failure can occur, and especially late-stage failure is quite a risk for manufacturers and vaccine developers. So if you go to the next slide. The way that Periscope contributes to this process and accelerating the clinical development is that by applying biomarkers at each stage of the development cycle of these vaccines, we provide more confidence in the mechanism of action of these vaccines. At the same time, controlled human infection studies in a phase 2 study and possibly in a phase 3 study can provide proof of concept and demonstrate that vaccines work, thereby reducing the risks of late-stage failure and hopefully leading to licensure of new pertussis vaccines. Slide, please. And this brings me to the acknowledgement slide, and I think as same as the previous speaker. So I'm presenting here on behalf of a lot of people, and so this was not possible without the participation of all of these partners and their enormous amount of work. And I'd also like to acknowledge the study participants and of course the funders for making the Periscope project possible. And that's it from the Periscope project.
Thank you very much, Dimitri, for this excellent presentation, and once again shows how a project can actually contribute to changes in practice, not only in the clinical trials, but also leading towards a closer toward the market licensing of vaccines. As has been said by our previous speakers, IHI is a public-private partnership, and I'm very happy to have with us today Laurence Torcel-Pagnol, who has worked for the past 10 years in European and worldwide public-private partnerships. Laurence Torcel-Pagnol is a global medical evidence generation lead for influenza at Sanofi Vaccines. She'll be joined by Dr. Javier Diaz-Domingo, who is the head of the Vaccine Research Department at FISABIO. FISABIO is the Foundation for the Promotion of Health and Biomedical Research of Valencia region in Spain. And both will present how DRIVE has designed an innovative platform whereby the industry members worked with public institutions in order to generate evidence for decision-making. The floor is yours, Laurence and Javier. Thank you very much.
Okay, thank you very much, Tech. Thank you very much for the IMI, IHI now for letting us do this important study. I'm Javier Diaz-Domingo, and Laurence will join me during this conversation. We are presenting an innovative platform for influenza vaccine effectiveness assessment, and we'll stick to the value of the collaboration across manufacturers and public sector. Next slide. We are focusing on influenza. Influenza is the infectious disease that has the highest burden of disease in Europe. And it is due because each season, there's nearly 11% of the population of the European population is affected by the infection. And though many people think that the flu is a mild disease, in fact, there's hundreds or thousands of hospitalizations due to influenza and up to 7,000 deaths per year in Europe, depending on the severity of the season. That brings that this is a very expensive disease that cost the Europeans nearly 40 billion euros of direct and indirect costs. And therefore, due to the burden of disease and the cost, there's a huge interest in Europe in public health to increase the vaccine coverage and also the companies to improve their vaccines. So we are having an increased number of vaccines with different technologies. And assessing vaccine performance, next slide, is very important. And Laurence is going to tell us about that.
Thank you, Javier, and hello, everybody. So yes, with this yearly vaccination, it's important to monitor vaccine performance in a real-life setting. And this is a joint mandate for Public Health Institute. We implement immunization program and guide health authorities and vaccine companies who have the obligation to continuously assess the benefits risk of their vaccine. But as we know, the real life environment is very complex, especially for influenza, and this is a huge challenge to provide quality data, robust estimate that account for the diversity and the heterogeneity observed in Europe on various circulation, vaccine technology, uptake, and population and settings. So next slide, please. So the genesis of DRIVE came from the change in 2016 of the IMI guidance on influenza vaccine. So this guidance requested a vaccine company to generate product-specific influenza vaccine effectiveness, and they encourage a company to liaise with institution and public health authority who had already infrastructure in place. So DRIVE was set up as a proof of concept of the IMA guidance with two objectives. Having a public-private multi-company partnership who answer to a regulatory commitment, so to test the governance and the regulatory pathway, and also to set up a platform to provide yearly brand-specific influenza vaccine effectiveness, and then to test the feasibility to get robust estimate for all ages, settings, brands in Europe, which is huge. So IMI was seen as the best mechanism to do this proof of concept. So we set up a 5 years project with 10 million euros in 2017 with 16 partners coming from 7 European countries. And with leveraging the work that has been done previously in an other IMI project that provided some nice output on a framework for post-marketing project. So next slide. So now, Javier, let's go to the key achievement of DRIVE. Let's start.
Okay, yeah. We managed to set up a large study platform in 7 EU countries in which we included hospitals, general practices, and also a population-based cohort that is continuously done in Finland. The importance of this platform is that we all use a common protocol, and therefore the results of the different sites could be easily pooled. Using a common protocol and using the pool analysis and using all the infrastructure that DRIVE allowed allow us to deliver the results 2 months after the end of the flu season. And also we had the possibility of increasing the health capacity due to the public-private funding. So during the 5 years that the study, take into account that we had the pandemic during these 5 years, and therefore we had 1 year with no flu, we managed to recruit over 35,000 of severe acute respiratory influenza, infections, and take or capture the impact of 13 influenza vaccines. So, for instance, in the year 1920, we managed to assess the influenza vaccine effectiveness with good precision of 3 of the vaccine brands that were in Europe. In the season 21-22, we managed to capture the value of 8 out of the 12 influenza vaccines that were in Europe. So we had an increase in capacity of assessing influenza vaccine effectiveness. And all the results, we were very open so we posted in our website the results of each season. And then we also had a good scientific communication. We had awareness campaigns about influenza. We had we participated in over 30 events. We wrote 26 newsletters and had 21 publications. But even as important as the achievements setting up this large platform was the development of the PPP governance model. That Laurence was the lead of this project. Laurence?
Yeah. So along the 5 years project, we have took a lot of energy to fine tune the governance of the platform and to get a transparent public-private partnership model. So we foster multi-stakeholder scientific collaboration. We organize an annual forum inviting also external stakeholders. We set up a transparent review process on the protocol and the result by all partners with an independent scientific committee oversight. So the study was conducted independently of vaccine company. But the scientific committee allow to have a mechanism that a company provided written feedback on the result, and the independent scientific committee adjudicated on that to support the result interpretation with public colleagues. And we also have a high quality data standard, as you can imagine, with this IMA oversight. So then what was also unique in this project is really to have a joint report with all brand-specific influenza vaccine estimates from all companies in the same document. And that has been submitted on a yearly basis to EMA. And each year, the vaccine working party from IMA did its evaluation and assessment. And along the 5 years, we had a really regular discussion with IMA that allow us to adjust the platform accordingly. So next slide, please. So despite the fact that in agreement with IMA, we focus on population with highest disease burden of influenza and relatively high coverage for the vaccine, it remains a challenge to get precise estimate for all the brand and to inform decision making. So in 2019-20, when IMA assessed our report, they deemed the data insufficient for decision making because they request to have a serial annual estimate obtained over a minimum of 3 years. And as you know, just after we had the COVID-19 pandemic that really stopped the, at least for the first years, the influenza virus circulation. So also along the 5 years, DRIVE faced some public-private partnership hesitancy and competition with a parallel public-only initiative that remain generally an obstacle to scale up the Drive platform. So as a conclusion, the platform was unique in offering consistently brand-specific influenza vaccine effectiveness results for each season at the European level. And this successful proof of concept deserves continuation, but needs clarity on the stakeholder role and responsibility. So currently there is a lot of discussion handled by EMA and ECDC regarding the future of that. So now let's move to Beyond Drive, Javier.
Yes, the Drive Steering Committee is still having meetings, periodical meetings, because we are really interested in the sustainability of the project, of this PPP project. So we can frame that in that we are encouraging data sharing practices. So all the Drive data is available for secondary use if anyone requests it under a protocol. And we, using the governance model of Drive, we managed to create the COVID Drive, which is the same philosophy, but instead of assessing the vaccine or the influenza vaccine effectiveness by brand, we are doing that with different COVID vaccines. This has been a very good achievement, and maybe we are moving towards new respiratory vaccines. And the discussions, as I mentioned, also are ongoing with the European Medicine Agency. And from the last meeting that we had is that they are using the lessons learned from Drive to they will take that into the revision of the next influenza guideline that the consultation is planned in the near future. Next one. So we have been representing a very large consortium with a good number of associated partners and research collaborations. Without them, this study would have not been able to conclude. You still can visit our website that will be running for the next year. And if you are more interested in our project, last year we published the end of the project publication, in which we recap all the benefits and hurdles of this project. So thank you very much to all the Drive community for this amazing effort that they did to achieve our results. And thank you very much for your attention.
Thank you.
Thank you both to Laurence and Javier for this excellent presentation and generating evidence against seasonal flu vaccine. I invite you to interact with our speakers now. Use the chat function below just to type in your question. I see that there are already a number of reactions and questions from the attendants. One question may be related to the last presentation that we've heard today, but it could also be addressed to all speakers today, is regarding data quality. So what the question is about is that unbiased data is critical, and this point has been quite challenged by public stakeholders as being dependent on industry. Is there any take on this that you would like to give on the data quality?
So yes, we have during the Drive, we understood that the possibility of people thinking that our results were driven by industry were very important. So we took a lot of firewalls for the industry to get into our data. So industry never got their hands into the data. So it was all the public part of the consortium that did all the analysis, they collected data, and then we wrote a draft or a preliminary script that was reviewed both by industry and by the independent scientific committee. Comments by the industry were non-binding. They were worthy but non-binding, and only those from the independent scientific committee were binding to us. So with this governance model, we ensure that the results were public, and the industry impact on these results was minimal. But definitely we had an important support, scientific support from industry apart from the budget.
Thank you very much, Javier. We have another questions regarding this public-private partnership hesitancy. The attendant would like to know what would be your recommendation based on your experience to move forwards towards improved collaboration between public and private. Laurence or Javier, once again.
Yeah, I can address this one. That's a big question. Thank you. So yeah, so I think what we need to make it clear is that there is two key components for public-private partnerships. The first one is transparency. You need to make really clear how you work together, who is doing what, to make sure that you have put in place appropriate boundaries, but also giving place for fruitful collaboration. And then the second is trust. And we know that trust is taking time. You know, when we starting, just to get you a sense, when we started Drive, you know, because having this first kickoff meeting all together, you know, so public and private, and also, you know, knowing that, you know, for company, we were all competitors in the market. So it's also putting all the company together. That was a huge, you know, challenge. But after the 5 years, we really succeed having this trust. And that's, for me, the big achievement that we got at the end. So then I think we need to pursue that and continue to promote collaboration by transparency and acknowledge that it's taking time. But I think we should not stop our effort. And I hope that in the future we can leverage further that.
Yeah, and I could add that from our learning is that joining efforts improves the results. Improves the results always with neutrality and with a lot of transparency. And I would, for those that PPP hesitancy is similar to the vaccine hesitancy. So they may need to learn how we are working. We may make more public of our governance model, because I think that reading our governance model and assessing it as we have done, the PPP hesitancy should decrease as it decreases vaccine hesitancy when vaccines are well explained.
Thank you very much, Javier and Laurence. Now a question for Ole and Dimitri. Although concerns over the harm and pain caused to animals in experimental studies and their physiological relevance are justified, do you think that non-animal-based systems have been developed to the point where they can fully replace the use of animals? Who wants to take it first? Ole.
Yeah, no, I think the short answer is no. But I think we need to see it as a journey. I mean, this we have to start where we can, and there's clearly in some cases a number of conditions where we cannot mimic cellularly or in vitro what happened in humans, or that we need animals. But this should not be, let's say, this should be an inspiration to continue rather than it should be holding us back. But I think it just demonstrates the importance of continued support to research, including basic research, to understand some of the basic cellular mechanisms and some of the immunological mechanisms that are underlying some of the biological processes where we now need animals to test, not at least for potency. I think is often the area where we cannot yet replace animals with in vitro testing. So we need for vaccines, we need to have much more basic knowledge about how the immune system works. What are the correlates of protection? What can we actually use as readouts in vitro tests before we can fully replace animals? So therefore, I think we should take what Vac2Vac and other projects colleagues here has achieved in the projects and use that as inspiration and really argue towards you, Tech, and your colleagues and policymakers that there's a need for sustained support and attention and funding for also the supporting the basic knowledge that is needed to really replace animal testing with non-animal test. So yeah, the short answer is no, but it's a journey that we are well underway towards replacing more and more animals, I think.
Thank you, Uli. And Dimitri, as you presented the human challenge model addressing this, what would be your take on this? How can we move forward from the human challenge model to this non-using animal testing any longer?
Well, I think like as Oli already mentioned, I think we're not there yet with like we're not at a stage yet where it can completely replace animal testing. But I am really convinced is that by moving to testing like vaccines actually in the target population in humans, we take away a translational gap that is there. And I think by using controlled human infection models, and not just for pertussis, but they're increasingly being used for other diseases as well, is that it is a fantastic opportunity is to use these models to learn more about what is necessary for protection, what are the biomarkers of protection, immunological correlates, but also to test new vaccines. And I think we are moving already like to a situation where new vaccines are being tested in this way, but also existing vaccines are being tested in this way. At the moment, there is still going to be need for animal testing at certain points in this process, but I have good hope is that in the future that might not be necessary anymore. But we're not there yet.
Thank you, Dimitri.
So I don't know if I could just add something to it. I fully agree with what Dimitri said, but in a way, you say what Dimitri and colleagues have been working on is almost replacing animals with humans.
We are
around, yeah. But just because we also have actually we are involved in another IHI project where also there are human challenge models as part of it, and I think, and I'm sure Dimitri will also agree to this, that a big part of it is also the regulatory acceptance of results in human challenge models. So there's also some work to be done there, have dialogues with regulators and understand how much is required from the different models for them to actually be accepted in the regulatory filing and so on. So a need for continued dialogue.
Yeah. No, it's absolutely correct, because these studies are not trivial, and there are many ethical aspects as well to consider. Yeah. And it's not like it's not a trivial study to do in terms of many aspects, in terms of like how to carry it out, but also in terms of like the regulatory aspects. But you have to keep in mind it is still a model. It is a fit for purpose model. It cannot fully replace field like phase 3 studies, I think in general, that's not how these vaccines, that's not how these type of studies work, but they do give direction, and they are being used more and more, like in also by industry to reduce risks. And I think that is already a good sign, because that also takes away the need to keep investing in vaccines that are simply not going to work.
To all speakers, the IMI2 vaccine projects have generated lots of interesting results. What do you think could be improved to provide further visibility and ensure that those results are generating further impact? The participants is thinking in terms of sustainability after the end of the project. We saw one part of the answer already in Drive, but in Periscope and Vac2Vac, what would be the sustainability after the end of the project?
Do you want to start, Ali, or should I start? So I can perhaps start. I mean, one of the things that we are doing with Periscope is because we have generated a large biobank of samples from all of these studies, is to make them available, and same as the assays that we are that we have developed for these immunoassays to measure the biomarkers of protection. And this is really critical in making them available to partners and to the worldwide global scientific community as well, because the higher the uptake of these assays, the more comparable things become, and I think also the more valuable the results become from future analyses. So that's something we're doing. I mean, we continue to provide output by publications, making data accessible, making the models accessible as well. I think that is really helping the sustainability of the project.
I can, from outside, I can only concur with this. I think what would be, let's say, a very down to earth help could be if there was some kind of means, and I'm not talking about big money, it's actually very modest money, to actually, after the end of the project, continue to support dissemination and advocacy efforts. I can take let's say, a very concrete example from our Vac2Vac project was that towards the end of the project, there had been a number of monoclonal antibodies developed, which we hope will be useful for, well, internally in the project, but also for the broader community to use for development of new non-animal models. So we came to the end of the project. It was very, we wanted to really make them available to the entire research community on an open basis, but we actually could not produce them within the project period and make them available. And we managed to solve that because the industry partners, mostly the industry partners, decided that they, in addition to the contribution to the project, also make an additional contribution financially to actually have them produced and make these monoclonal antibodies freely available through NIBSC in the UK. So I think but these kind of things is not very costly, but if industry partners not have stepped up, that would probably have fallen apart. But it now has a huge impact because now these antibodies are freely available to the entire research community. The same with I mean, publications that cannot be in principle be paid for after the end of the projects. Advocacy and dissemination, in our case, some of the partner organizations, not at least IABS, has made a major effort to really engage globally and promote, let's say, the results of Vac2Vac, but Europe in more general terms in this area. And it's not something that costs a lot of money, it's quite small money. So I think it would be very beneficial if there was some kind of funds or being available to these kind of dissemination efforts and sustainability after the official end of the project. but that's just an idea.
And maybe just to piggyback on those comments, I think like I recognize what you say, but also biobanking itself.
Yeah.
It's like, so we established biobanks, large biobanks. So there's a huge investment in getting these studies funded and doing them, and then what usually happens at the end of these projects, there are samples remaining, and that are of really high value to the community. But now it's basically in these projects, it is more difficult to maintain such a biobank because the partners, and we have decided to do this in a central location, is basically have to fund that themselves. So we're now getting finding methods is to share the cost for maintaining that biobank. But I think that would be very useful for IMI in general. It's like if you provide such a if you do an investment in all of these studies, I think it's also useful to also at the end is to say, okay, but what will happen with these samples, and can we actually make sure is that they are used to the best of our options.
If I may add, at the end of Drive, it was quite a challenge to pursue all the effort, and I join what you are saying, because for us, for example, it took 1 year to set up this secondary use framework to leverage all the data that has been collected along the 5 years. And it's true that as soon as you stop the IMI official project, you are out of any umbrella. So you have no more funds officially and no more legal environment. So that's why it's taking time, efforts. And of course, it's something that maybe should be improved for the future. That piece that should be, because in the project you have this sustainability component, but it's the topic is there, but all the the environment, the legal and financial environment that is needed to pursue that, it's not really concretely there, and that's sometimes taking time to set up that.
Indeed. Thank you very much for your answer. Hearing what you said, you were talking about the value of the biobanks, the different assays that you have collected, and the experience that you have gathered through this, the duration of the different projects. What would you say remain the unmet needs that you would like to tackle based on the experience that you have so far? Dimitri, Ole, and Javier, Laurence. Any unmet need that you can think of?
So, well, from the Periscope consortium, I think like the unmet need is that so we were initially restricted to analyze new vaccines concept, because it was a pre-competitive project, so we had to focus on licensed vaccines. I think that is now changing, but it would have been very useful to also analyze new vaccines, because they're also based on completely new mechanisms of protection. And so that would be very useful. I think we're moving there with the controlled human infection models, but in terms of the assays, I think applying these assays to other vaccines is already quite useful. But basically with the current way that we were doing this, we had to focus on the acellular and the wholesale vaccines that were already on the market. But I understand why it was done. But it would still be useful to focus on these other vaccines as well.
Thank you. Ole, you want to take?
No, I don't have so much more to add. I think just, I mean, if you save the unmet need, I think I mean, for many of the things we are doing, it's something, it takes time. Things takes time, and I mean, in the VAC to VAC, and I think that applies probably to many projects. it's like a big train. It takes a long time to get it rolling. And then once it's up and rolling, you generate results, and then it's like you are in top speed, but then you've already reached the end station, which is often a pity because it's after that you often have, you could continue and you could produce a lot of important results. So I think, of course, this, and that's, of course, this should not be an automatic extension and things like that, but often it's a matter of, I mean, many results are generated towards the end of the project, and therefore there's not much time really to establish, let's say, a viable sustainability plan within the project period, because what you need to really establish the sustainability only comes towards the end of the project. So I think that's again a little bit the sort of the longer term perspective of many of these things. How can we keep it going? But of course, there's no easy solution to that. But I think the first step could be things like, if there were some general funds available to keep biobanks going, or keep, I mean, in our case, we had also a type of biobank, I guess, but the monoclonal antibodies, making them available. So these kind of things, and also, of course, dissemination efforts and really spreading the word. So if there were some kind of mechanism to assure that that could at least be maintained, I think that would be already a huge gain for everybody.
Thank you. Javier or Laurence, any take on the unmet needs that you'd like to tackle?
Yes, I could like to address a couple of things. First of all, is that PPPs in all research areas, not only in health, is heavily supported by the European Commission. And it seems that it's quite very welcome by the population in all areas except in health. If we see the health world, the industry is developing their own products. Then, in the case of vaccines, the public health is unhappy of assessing the effectiveness. So it seems that there's a blockage between I'm developing a vaccine or industry is developing a vaccine, and then we do not allow the the industry to evaluate its own vaccine. We are in a societal change. We are in the 21st century, and there's no studies or big reports on PPP hesitancy. So I think that a need is to do good sociological studies to assess, first of all, the impact of vaccine hesitancy or PPP hesitancy, and second, how to overcome that with clinical trials or whatever. So PPP is a real need, but we should tackle PPP hesitancy. And to tackle that, it is important to know what's happening. Over.
Thank you.
So if I may complete, I agree with Javier, and I think what is important to acknowledge is that you have a gradient of sensibility. You know, when you are in the research area, it's quite safe, public-private together. And then when you move, and this is what we have observed, because we started, I will say, this long story with advanced IMI project, which were, you know, on methods for post-marketing settings. And then when we move with Drive, we really true product and post-marketing environment, everybody was really, you know, feel, you know, uncomfortable at the start. And in fact, for us, it's really a big question mark, because when we look at the vaccine development company submit data for registration and on their own. And even if we know that observational study need to be carefully developed, because we know that there is a lot of bias, when we were all expert scientists together, public and private colleagues in Drive, we all agree on the methods. We all agree on the way to manage bias and so on. So at the end, the results are the one that, of course, in real life setting, you can trust with knowing the environment and the difficulty. But for me, it's very important to understand the mechanism and how you reach out this kind of collaboration and scientific output at the end.
Thank you very much. And with that, would like once again to conclude this IHI impact series on vaccine. Thank you to all our distinguished speakers. That was excellent result that you have presented. And see you very soon for the next step in the vaccine development. Thank you and wish you a nice day.
Thank you very much indeed. Bye.
Well,
Thank you.
Bye-bye.
Bye-bye.